This is my protein KRAS, as you can see, and I have shown that with bendy straws and pipe cleaners and pieces of candy. So KRAS is localized in the plasma membrane of a cell, since it is a key signaling molecule. It is found on the chromosome 12 in mammals, such as human and mice. And like I said, it's present in the cell membrane and in the cytosol of a cell because it is important in relaying messages to the nucleus. This is the overall 3D shape of my protein KRAS. I represented it with alpha helixes and beta sheets, which you can see alpha helixes are this pipe cleaner, and my beta sheets are represented by this bendy straw, which represents the planar of beta sheets. And I also have my KRAS molecule broken down into different domains. It is one singular subunit, so you can see that because it is all connected, and it is very small subunit usually. However, it does have our different domains, and that would be like the G domain of it, which helps play a role in the binding of GDP, which would be down here in my KRAS molecule. And then the other key subunit of this molecule is the HVR, which is the hypervariable region, which basically this just helps anchor the protein and is what allows us to have the signaling molecule, since it keeps the protein anchored. And most proteins do have this region, or many HVRs. Like I was talking about previously, KRAS has our alpha helixes and our beta strands or sheets within this G domain and the other domain, and this means that our protein would be characterized as a secondary structure, and you can see that also in my model. Okay, so I am now going to show how my assigned mutation of KRAS affects the actual structure. So as you can see here, this is a non-mutated KRAS, so it's very flexible in movement, and my P site or P loop is bound to GDP loosely, so it is not always locked and in control. And same with my switch 1 and 2 strands. They are very conformational and can change easily, which we see in a normal KRAS protein. And then I will show the mutated KRAS. So this is now my mutated KRAS protein, which the mutation occurs in glycine 12, and that's a switch from glycine to cysteine usually. It can also be many other amino acid switches, but it's always usually located in glycine 12, which we see that shift from glycine to cysteine in a thiol chain, side chain, which is why we see more locked interactions in the KRAS mutated form. So I represented this by showing that my P loop is in a locked state, and I represent this by a bound GTP, meaning it's stuck in continuing cell proliferation, and also by just a significant piece showing that we basically no longer have glycine. It is now cysteine, or another bulkier amino acid that causes these stuck interactions, and also showing that my switches are no longer conformational and can change easily when KRAS is mutated. So basically the stuck conformation of KRAS and mutation is why we have continuous cell proliferation, which eventually will lead to cancer. It's just an overgrowth of cells. 3 limitations that I think my model represents is the beta sheets are not as planar as I would like them to be. My P-loop, I wish I could represent this better and show the GTP activation, and also showing my HVR section.